if you break your arm, I mean, an X-ray gives you a clean binary answer. You look at that jagged white line on the film, and it's either broken or it's not.
Right. It's very straightforward.
Yeah, but for you, stepping into the world of molecular medicine and, human physiology, you already know that most human disease doesn't look like a clear-cut fracture.
No, not at all.
It looks like a single misfolded protein deep inside a muscle fiber or a missing chemical shipping label on a microscopic enzyme. The diagnostic landscape at this level isn't a crisp X-ray. It's incredibly murky.
It's a completely different paradigm. At the biochemical level, you aren't dealing with macroscopic anatomy. You're dealing with invisible physical forces, kinetic rates, molecular geometry. The pathology is driven by molecules colliding in a solution.
Which is why mastering those microscopic collisions is really the only way to actually see the whole picture of human health.
Yeah.
You need that comprehensive, high-yield mastery of biochemistry without getting, lost in the textbook weeds. So today, we're taking a deep dive into the USMLE Roadmap Biochemistry to map out that landscape. Our mission is to extract the core physiological chemistry, the metabolic pathways, and the genetic blueprints that run the human body.
And crucially, we want to directly tie every single one of those molecular mechanisms to clinical applications.
Right. Okay, let's unpack this. We have to start with the environment itself, because all of these complex metabolic reactions happen in a solvent, and that solvent is, well, water.
Yeah, we tend to take water for granted, but chemically, its hydrogen bonds are doing a massive amount of physiological heavy lifting.
It's crazy when you think about it.
It really is. The unique physical properties of water dictate our very survival. Take its incredibly high heat of vaporization, for example.
Okay, so that means it takes a lot of energy to boil it.
Right, because water molecules are so tightly hydrogen bonded to one another, it takes a massive input of thermal energy to break those bonds and convert liquid water into a gas.
And in human physiology, this translates to the cooling mechanism of sweat, right?
Exactly. As perspiration evaporates from the skin, it literally rips that massive amount of heat energy away from the body. It prevents hyperthermia during heavy exertion.
But those same tight hydrogen bonds, they create a major mechanical nightmare inside the lungs, specifically regarding surface tension.
Yeah, surface tension is the cohesive force pulling water molecules together at an air-liquid interface. Down in the microscopic alveoli of the lungs, a thin layer of water coats the tissue.
And those hydrogen bonds are constantly trying to pull together, right?
Yeah, generating a collapsing force on those tiny air sacs. To counter this, type 2 pneumocytes secrete lung surfactant,
Which is like a mix of proteins and lipids.
specifically amphipathic lipids molecules with a hydrophilic head and a hydrophobic tail.
So they act as a chemical wedge. They slip into the water layer, positioning their hydrophobic tails in the airspace, and physically disrupt that hydrogen bond lattice.
Yes, that physical disruption drastically reduces the surface tension. It increases lung compliance so the tissue can easily expand when you inhale.
And clinically, this is a huge deal for premature babies.
Huge. Infants born before about 35 weeks often lack sufficient surfactant. They develop respiratory distress syndrome.
Because without that chemical wedge, the water's surface tension is just too strong.
Exactly. The alveoli collapse, and the infant's respiratory muscles quickly fatigue trying to force them open. The clinical intervention relies on administering maternal corticosteroids to accelerate fetal lung maturity or directly instilling exogenous surfactant into the airway.
Wow. Okay, so the environment isn't just about water molecules, though. It's about the hydrogen ions floating in that water. The body maintains arterial blood pH in an incredibly narrow window around 7.4.
Very narrow. And to survive metabolic shifts, we rely on buffers, which act like cellular shock absorbers, basically soaking up excess acid or base.
Right, and the mechanics of buffering are described by the Henderson-Hasselbalch equation, which says that a weak acid provides the maximum buffering capacity when the surrounding pH matches its own pK.
That's the core principle, yes.
But okay, I have to push back on this because the math never seemed to make sense for human physiology.
How so?
Well, if our survival depends on keeping blood pH strictly at 7.4, why is our primary buffering mechanism the carbonic acid bicarbonate system? Its pKa is only 6.1. A pK of 6.1 mathematically makes it an absolutely terrible buffer for a target pH of 7.4.
What's fascinating here is that you're totally right. If we were talking about a closed beaker on a laboratory bench,
Okay.
in a closed system, it would fail instantly. But the human body is an open system. The carbonic acid in your blood is in constant dynamic equilibrium with dissolved carbon dioxide gas.
Oh, I see where this is going.
Yeah, so if a patient develops metabolic acidosis,
Yeah.
say they're in hypovolemic shock, generating massive amounts of lactic acid, the excess protons bind to bicarbonate, forming carbonic acid.
Which then instantly dissociates into water and CO2.
Right, and because it's an open system, the brainstem detects this and triggers hyperventilation. The patient breathes rapidly and deeply, physically expelling that CO2 into the atmosphere.
So by continuously removing the product of the reaction, the body forces the chemical equilibrium to keep shifting, absorbing more and more dangerous acid.
Exactly. The respiratory compensation literally blows the acid out of the body. It's a brilliant workaround.
That is wild. And that tightly regulated environment allows the actual workers of the cell, the proteins and enzymes, to function.
Yes, and we know amino acid chains fold into complex three-dimensional shapes based on their side chains, but the clinical implications of that folding are profound. Form absolutely dictates function.
Right, like with collagen.
Exactly. Collagen is the most abundant protein in the human body. It provides the tensile strengths for skin, tendons, and bones, and it requires a highly specific quaternary structure, a tightly wound triple helix.
And to pack 3 polypeptide chains that closely together, every third amino acid has to be glycine.
Yes, simply because its side chain is just a single hydrogen atom. Anything larger causes steric hindrance, which prevents the tight wind.
Which perfectly explains osteogenesis imperfecta, or brittle bone disease. A genetic mutation swaps out that tiny glycine for a bulkier amino acid, the helix fails to pack, and the bones lose their structural integrity.
Precisely. And even if you have the glycine, you still need to lock that helix together with hydrogen bonds, which requires hydroxylating the proline and lysine residues.
And that hydroxylation strictly requires vitamin C, doesn't it?
It does. It acts as an enzymatic cofactor. A deficiency in vitamin C halts this modification. The triple helices remain loose and unstable, leading to scurvy.
Which causes the bleeding gums and poor wound healing because the collagen framework is just falling apart.
Right. Structure dictates everything. Let's look at oxygen handling. Myoglobin in the muscle and hemoglobin in the red blood cell both bind oxygen, but their structural differences completely change their behavior.
Right, they have very different jobs.
Yeah, myoglobin is a single polypeptide monomer. It has an incredibly high affinity for oxygen, meaning it grabs it and refuses to let go until the muscle is under severe metabolic duress and tissue oxygen levels plummet. It's a storage vault.
Whereas hemoglobin is a tetramer, it has 4 distinct subunits.
Exactly. Its evolutionary job isn't storage, it's delivery. It must efficiently bind oxygen in the pulmonary capillaries where it's abundant, but more crucially, it must readily surrender that oxygen in the peripheral tissues where it's needed.
You know, I always visualize hemoglobin cooperativity like an empty bus on a rainy day.
Oh, I like that.
Yeah, when the bus is completely empty, it's remarkably hard to convince that first passenger to get on. But the moment the first person boards and sits down, it changes the dynamic. It becomes exponentially more attractive for the next 3 passengers to jump on board.
That's a great way to think about it. At a molecular level, when that first oxygen molecule binds to one heme group, it physically pulls on the histidine residue attached to it.
Which changes the shape of the whole molecule.
Right. This structural tug breaks stabilizing salt bridges, triggering an allosteric conformational shift across the entire tetramer. The molecule transitions from a tense state to a relaxed state, massively increasing the remaining subunit's affinity for oxygen.
But the clinical brilliance is how the body forces hemoglobin to empty that bus when tissues are starving for oxygen, like when you rapidly ascend to high altitude.
Yeah, at high altitude, atmospheric oxygen pressure drops, threatening tissue hypoxia. The red blood cells respond by rapidly synthesizing a molecule called 2.3-bisphosphoglycerate, or 2.3-BPG.
And what does that do?
It wedges itself into the central cavity of the hemoglobin tetramer, physically cross-linking the beta subunits. This locked the hemoglobin in its tense low-affinity state, forcefully promoting the release of its oxygen payload into the tissues.
To compensate for the hypoxic environment, it's an automatic molecular altitude adjustment.
Exactly.
Now, when we talk about enzymes, which are just, specialized catalytic proteins, understanding their kinetics is basically the foundation of pharmacology. We deal with Vmax, the absolute maximum velocity of the enzyme, and Coulomb, which represents the affinity for its substrate.
Right. A low Coulomb indicates a high binding affinity. The enzyme eagerly snaps up the substrate, even at very low concentrations, and we exploit these kinetics pharmacologically all the time.
Like with statin drugs.
Yes. Statins, used to lower cholesterol, are competitive inhibitors. Their molecular structure mimics the natural substrate of HMG-CoA reductase. They physically block the active site.
But because it's a direct competition for the same parking spot, you can overwhelm the drug just by flooding the system with massive amounts of the original substrate, right?
Exactly. The drug increases the apparent quorum, making it seem like the enzyme has lower affinity. But if you add enough substrate, the enzyme can still reach its normal Vmax.
But non-competitive inhibitors are different.
Very different. They don't bother fighting for the active site. They bind to an allosteric site elsewhere on the enzyme, inducing a conformational change that distorts the active site entirely.
So you could flood the system with all the substrate in the world, but the enzyme is just physically broken?
Right. The Vmax plummets while the NDEV remains unchanged because the active sites that aren't inhibited still bind the substrate normally.
And clinicians rely on tissue-specific enzymes, that catalyze the same reaction but have slight structural variations depending on where they live, like creatine kinase or CK.
Yeah, the classic example. The heart specifically expresses the CKMB isozyme. If a patient presents to the emergency department with crushing chest pain, detecting elevated CKMB in their serum provides biochemical proof of a myocardial infarction.
Because the cardiac myocytes have necrotized, ruptured, and spilled this specific isozyme into the circulation.
Precise.
Speaking of membranes rupturing, controlling what goes in and out of the intact cell is a massive metabolic undertaking. The boundary is an amphipathic lipid bilayer, heavily regulated by cholesterol to maintain optimal fluidity.
And embedded within this sea of lipids are transmembrane proteins acting as highly specific gates and pumps.
Right. Facilitated diffusion via uniport carriers allows molecules like glucose to enter cells passively down their concentration gradient.
Yeah, the glucose binds to the extracellular phase of the transporter, triggering a conformational ping-pong shift that closes the outer gate and opens the inner gate, releasing the sugar into the cytoplasm.
But maintaining the electrical gradients that allow neurons to fire and muscles to contract requires active transport. The sodium-potassium ATPase pump is an absolute energy hog.
Oh, massively.
It actively burns a molecule of ATP to force 3 sodium ions out of the cell and drag 2 potassium ions in, constantly fighting against their natural concentration gradients.
It's actually estimated that at rest, nearly a quarter of your body's entire ATP expenditure is dedicated solely to fueling that single type of membrane pump.
A quarter? That's insane. But here's where it gets really interesting regarding the lipids that make up those membranes and the fat we consume.
Okay, let's hear it.
The hydrocarbon tails of these lipids can be saturated or unsaturated. We know saturated fats, which are solid at room temperature, are linked to atherosclerosis. Unsaturated fats, like olive oil, have double bonds in their tails, keeping them liquid and generally healthier.
Right.
But then we have artificial trans fats. Trans fats have double bonds, so they are technically unsaturated. Shouldn't they act just like healthy vegetable oils in the body?
You would think so, but the danger lies purely in the physical geometry of the molecule. Natural unsaturated fats contain cis double bonds. This specific chemical configuration creates a rigid 30-degree kink in the hydrocarbon chain.
So they can't pack tightly together.
Exactly. Because of that sharp bend, they remain fluid oils. But industrial hydrogenation creates trans double bonds. This alters the geometry, making the hydrocarbon chain completely linear, structurally mimicking a saturated fat.
Ah, because they are straight, they pack densely together, forming solids like shortening?
Yep. And inside the vascular endothelium, they aggregate exactly like saturated fats, aggressively driving LDL cholesterol elevation and arterial plaque formation.
It's a structural illusion that totally bypasses our metabolic defenses. But, maintaining that cellular defense, powering that sodium-potassium pump, synthesizing those complex proteins, it's exhausting.
It requires a constant supply of fuel.
Right, which brings up the ultimate physiological question.
Yeah.
How does the cell constantly manufacture the vast amounts of ATP required to stay alive? It boils down to the hormonal tug-of-war regulating the fed and fasting states.
In the fed state, a carbohydrate-rich meal spikes blood glucose. The pancreatic beta cells secrete insulin, the body's master anabolic hormone.
And insulin doesn't just passively suggest the cell take up glucose, right?
No, it orchestrates a massive architectural shift. It triggers an intracellular kinase cascade that recruits vesicles packed with GLUT4 transporters, forcing them to fuse with the cell membrane. The cell suddenly sprouts thousands of new glucose channels, vacuuming sugar out of the bloodstream.
And once inside, insulin activates the enzymes for glycogenesis, physically stitching that glucose into dense glycogen granules in the liver and muscle for later.
And once those storage tanks are full, it shifts the metabolic machinery toward lipid synthesis, converting excess energy into triglycerides packed into adipose tissue.
But as hours pass and you enter the fasting state, insulin levels plummet and glucagon takes the helm. Glucagon is strictly catabolic.
Right. It binds to hepatic receptors, activating cascades that violently break down those glycogen stores, glycogenolysis dumping free glucose back into the blood to sustain the brain.
And once the glycogen is depleted, glucagon forces the liver into gluconeogenesis, literally synthesizing brand new glucose molecules from amino acid skeletons.
And if you transition from fasting to prolonged starvation, cortisol takes over. Cortisol aggressively breaks down skeletal muscle to supply amino acids and triggers lipolysis, liberating free fatty acids from adipose tissue.
But wait, the brain, which is the body's biggest energy consumer, it can't utilize fatty acids because they can't effectively cross the blood-brain barrier.
That's true. And this is where the liver performs a vital metabolic pivot. It takes those circulating fatty acids and oxidizes them into ketone bodies, specifically acetoacetate and 3-hydroxybutyrate.
And those can cross the barrier.
Yes, these water-soluble molecules easily cross into the brain, providing a crucial alternative fuel source and sparing the body's rapidly dwindling protein reserves.
That's amazing. Let's look under the hood at how a cell actually extracts energy from that glucose, starting with glycolysis. The very first step is trapping the glucose inside the cell by attaching a phosphate group to it.
Right, which relies on hexokinase in most tissues.
And hexokinase has a very low collimeter. It's incredibly selfish, it has a high affinity, and will rapidly grab any available glucose to ensure the cell's own immediate survival.
The liver, however, uses the glucokinase isozyme, which has a very high collimeter. It has low affinity and actually ignores normal fasting blood glucose levels.
So it only turns on when glucose is super high.
Exactly. It only activates after a massive meal when glucose concentrations are extremely high, allowing the liver to act as a metabolic sponge, sequestering the sheer excess to prevent dangerous postprandial hyperglycemia.
So glycolysis breaks that 6-carbon glucose into 2 3-carbon pyruvate molecules, netting 2 ATP. But it also strips high-energy electrons off the carbon skeleton, loading them onto a carrier molecule called NAD+ to form NADH.
Yes, and if the cell is sprinting and oxygen is scarce, those NADH carriers can't drop their electrons off in the mitochondria.
The cell quickly runs out of empty NAD+ carriers and glycolysis stalls.
Right, so to survive anaerobically, the cell dumps those electrons directly onto pyruvate, converting it into lactate.
Which regenerates the NAD+, so glycolysis can continue, but the rising lactate drops the tissue pH, causing acute muscle fatigue.
Now, if oxygen is plentiful, that pyruvate enters the mitochondria and is converted into acetyl-CoA, which is the primary fuel for the TCA cycle.
I always think of the TCA cycle exactly like a massive biological water wheel. You dump the carbon skeletons of acetyl-CoA into the top. As the enzymatic wheel turns, it crushes the carbon bonds, releasing CO2.
But the actual purpose of the wheel isn't to make ATP directly.
Right. It's designed to extract a torrential flow of high-energy electrons, capturing them on NADH and FADH2, and funneling them toward the cell's main power generator, the electron transport chain.
The electron transport chain is a series of massive protein complexes embedded in the inner mitochondrial membrane. As NADH and FADH2 deliver their electrons, these complexes pass the electrons down a localized electrical wire.
And they use that energy to pump protons, right?
Exactly. They use the energy from that electron flow to physically pump protons out of the mitochondrial matrix, creating a massive electrochemical gradient. It's like a biological dam holding back a reservoir of highly concentrated protons.
And the grand finale is ATP synthase. The protons are allowed to rush back through this transmembrane channel, acting like a rushing river spinning a turbine.
Yes. The physical rotation of the ATP synthase motor literally crushes ADP and inorganic phosphate together, generating the overwhelming majority of the ATP that keeps us alive.
But physiology always has a backup plan. The body can intentionally sabotage this system. Thermogenin, an uncoupling protein highly expressed in the brown adipose tissue of human infants, embeds itself in that mitochondrial membrane.
It creates a pore that allows the protons to bypass ATP synthase entirely.
So the gradient dissipates, but the energy isn't captured as ATP. It is explosively released as pure thermal energy. It literally burns cellular fat to generate heat, protecting the infant from lethal hypothermia.
It's an incredibly elegant survival mechanism. But, every single pump, receptor, and enzyme we just discussed is completely useless without the instructions to build them. That's where the nucleus takes over.
Right.
The central dogma dictates that DNA is transcribed into RNA, which is then translated into functional proteins. But before a cell can divide, it must replicate its entire genome with staggering speed and precision.
And DNA polymerase III is the primary engine here. It clamps onto the unwound DNA strand and synthesizes the new strand at a rate of nearly 1,000 nucleotides per second.
But at that kinetic speed, errors are inevitable. If a wrong nucleotide slips in, it could trigger a lethal mutation.
Which is why DNA pull-through III has that built-in backspace key, right?
Yeah.
The 3 to 5 exonuclease proofreading activity.
Exactly. The instant it senses a mismatched base pair, altering the width of the double helix, it halts, reverses direction, snips out the error, replaces it with the correct nucleotide, and resumes sprinting. That exonuclease activity is the only thing standing between us and catastrophic genetic failure.
Wow. And once transcribed and translated, many proteins require intense post-translational modification in the Golgi apparatus to function or reach their proper destination.
The Golgi acts as the cell's shipping department. For instance, enzymes destined for the lysosome, the cell's acidic recycling center, they must be tagged with a specific molecular barcode.
A mannose 6-phosphate residue.
Precisely.
And if a genetic defect breaks the enzyme responsible for attaching that tag, the shipping department fails. The lysosomal enzymes are mistakenly secreted out into the extracellular space.
And inside the cell, the lysosomes become engorged with cellular debris they can no longer degrade. This leads to eye cell disease, which is a devastating and fatal childhood metabolic disorder.
That's heartbreaking. And while we typically think of genetics in Mendelian terms, dominant and recessive alleles inherited from both parents, mitochondrial DNA operates under a completely different paradigm.
Yes, it is inherited exclusively from the maternal lineage, as the ovum provides all the organelles for the developing zygote.
But this always raises a fundamental clinical question. If a mother harbors a mitochondrial DNA mutation, she passes those exact same mitochondria to all of her children. Why, then, would one child suffer from devastating neurological deficits while their sibling is entirely asymptomatic?
If we connect this to the bigger picture of embryonic cell division, we see the phenomenon of heteroplasmy.
Heteroplasmy.
Yes, a single cell contains hundreds of mitochondria. In a carrier mother, there is a mixed population. Some mitochondria contain wild-type DNA, while others harbor the mutant genome. During oogenesis and early embryonic cleavage, these mitochondria are segregated utterly at random into the daughter cells.
Oh, I see. So one embryo might inherit a threshold of 90% mutant mitochondria in its developing neural tissue, leading to severe pathology?
Exactly. And another embryo might inherit only 10%, remaining phenotypically normal. The clinical severity is a direct result of this microscopic random genetic lottery.
Which brings us to the ultimate regulatory challenge. Controlling when a cell is allowed to utilize these blueprints to divide and grow. Cellular signaling is the tightest control center in physiology, and when those signaling cascades break down, cancer is the inevitable result.
Cells are heavily dependent on external authorization to divide, primarily mediated through transmembrane receptors like receptor tyrosine kinases. When a growth factor binds the extracellular domain, the receptor autophosphorylates its intracellular domain, initiating a massive cascading chain reaction of kinases.
And the most notorious relay switch in this cascade is the RAS protein. RAS operates as an oncogene. Under normal conditions, it binds GTP to flip into the active on state, transmitting the signal to divide.
Critically, it possesses intrinsic GTPase activity, a built-in timer that automatically hydrolyzes the GTP, flipping the switch back off.
But highly specific point mutations strip RAS of this enzymatic timer. It becomes permanently locked in the active conformation, flooding the nucleus with relentless division signals.
And the body's defense against this hyperactive signaling relies on tumor suppressor genes. These act as the ultimate molecular checkpoints, sensing DNA damage or aberrant signaling, and actively halting the cell cycle to allow for repair.
I look at an oncogene like a broken thermostat in a factory. It's permanently sending the build signal, flooding the assembly line. But the tumor suppressor gene is the quality control inspector.
That's a good analogy.
Even if the factory is in overdrive, the inspector can hit the emergency shutoff if the products are defective. Because we inherit two alleles, you have two inspectors. If a mutation knocks out one allele, the remaining inspector can still manage the line.
But if a cell suffers a loss of heterozygosity, mutating that second allele, The inspector goes on strike.
The thermostat is broken, quality control is gone, and the cell plunges into unregulated malignant proliferation.
And in a healthy cell, that level of catastrophic damage activates the intrinsic apoptotic pathway. The mitochondria, sensing the unrepairable state of the cell, permeabilize their outer membrane. They leak cytochrome C into the cytoplasm, a biochemical point of no return.
And what does that do?
Cytochrome spec nucleates a complex that activates caspases. These are proteolytic enzymes that systematically and deliberately dismantle the cell's cytoskeleton and shred its DNA from the inside out, silently sacrificing the individual cell to protect the integrity of the organism.
What an incredible journey we've mapped out today. We started with the simple physics of hydrogen bonds allowing an infant's lungs to inflate, built up through the allosteric machinery of hemoglobin delivering oxygen, watched the TCA cycle physically crush carbon bonds to generate the electricity of life, and finally decoded the intricate signaling checkpoints balancing cellular life and death.
It really is a massive interconnected web.
It is. So what does this all mean for your clinical mastery? Let's put you to the test with a rapid-fire review question to synthesize these concepts. If a patient presents in the emergency department hyperventilating and their labs show a massively elevated blood lactate level, is their blood pH likely acidic or basic? And structurally, what exactly is their body attempting to do with that carbon dioxide?
Well, the lactate accumulation indicates profound metabolic acidosis, meaning their blood pH is severely acidic. By triggering hyperventilation, the brainstem is forcing the lungs to blow off CO2. This physically removes the product of the carbonic acid by carbonate buffer system, constantly shifting the chemical equilibrium to pull more and more toxic hydrogen ions out of the circulation, dragging the pH back toward a survivable baseline.
It is a testament to the staggering interconnectedness of human biochemistry.
Absolutely. And as you continue to study these pathways, consider this final thought. We spent a lot of time analyzing the massive dynamic complexity of hemoglobin. Yet a single point mutation in the genetic blueprint, swapping out just one single nucleotide to replace a glutamate with a valine residue, is all it takes to drastically alter the physical geometry of that entire tetramer. It causes the hemoglobin to rapidly polymerize and warp the red blood cell into a sickle shape, resulting in sickle cell anemia.
One single nucleotide.
Just one. It is a stunning, sobering reminder that in the murky world of molecular medicine, the most microscopic change in the code can irrevocably alter the destiny of the entire human organism.
Keep exploring, and we'll see you on the next deep dive.